Since bacteria all have restriction endonucleases to combat invasion by foreign DNA, a mechanism to protect their own DNA is essential. A clever solution to this problem of a restriction nuclease cutting its own DNA is to methylate DNA at each target site sequence using a partner restriction methylase. The methyl groups distort the restriction endonuclease's active site and inhibit its activity so that DNA is not digested. This modification therefore protects the bacterial DNA from digestion by its own endonuclease and is introduced during the replication process, preventing digestion of the organismal DNA. This pairing of methylase and nuclease provides a single-celled organism exposed to DNA in the environment a mechanism to both digest invading foreign DNA and to protect its own DNA. As we have discussed, restriction endonucleases, often just referred to as restriction enzymes, have a wide range of target sequences so that when digestion of a DNA sample occurs, there are multiple fragments. Because the restriction sites are short, there are always multiple fragments when you digest a DNA with a restriction enzyme. So that the product of the digestion is a mixture of DNA fragments with the same ends, because the same site was recognized, but the fragments differ in length. Working with this mixture is problematic, so separation is a key issue, and gel electrophoresis is a ready solution with separation by size because DNA has a charge. If you recall protein separation on SDS gel electrophoresis, the SDS is used to denature and bind to the protein to introduce a uniform charge mass ratio. But that uniform charge mass ratio is built into DNA with 2 phosphates for every base pair, allowing separation of DNA fragments by length. Think briefly about which moves fastest, shorter or longer fragments. The DNA can be identified by dye, radiolabel, or hybridization after denaturation and probing with a cognate DNA. Note that the shortest DNAs move most rapidly, the longest DNAs move slower, just as we observe for proteins in SDS gel electrophoresis. And you can see that in the gel shown on the right, the way in which staining can occur using a fluorescent dye that can be seen in UV light. One way this type of separation of restriction fragments was initially used was in comparing DNA samples from different sources. Restriction sites are not uniform within a species, so individuals have different DNA sequences and therefore different sites and therefore different lengths of restriction fragments that can be compared after digestion and separation, allowing what has become known as DNA fingerprinting or restriction fragment length polymorphism, RFLP analysis. Taking DNA from different individuals, humans, trees, animals, soil bacteria, any source of DNA, and treating with a restriction enzyme yields a fingerprint that can be compared between individual samples. Sometimes samples are amplified by PCR prior to analysis, but that's done very carefully to avoid contamination. Fragments are separated by gel electrophoresis and analyzed by staining or by transferring onto a membrane and probing with specific nucleotide sequences. The fragments are visualized by fluorescent probes, by radiolabel, targeted to specific sequences, and images are created on film or an electronic format. This is a DNA fingerprint using RFLP analysis. This method quickly became fodder for television crime shows, and indeed this approach remains widely used in the forensic setting. Because it is possible to take DNA from a crime scene or any other setting and compare the fingerprint of the DNA, the pattern of bands, the RFLP pattern, to individuals suspected in the specific crime or to another organism or another species, this comparison allows many applications from identifying paternity to applications in evolutionary biology. Now, DNA sequencing, as we discussed in last week's homework, has provided a more detailed analysis of DNA starting in the 1970s. but it has advanced rapidly since that time with multiple so-called next generation or next gen methods to garner DNA sequences rapidly and accurately. DNA sequence data allowed deduction of amino acid sequence and therefore comparison of protein sequences. In addition, sequences of non-coding regions, including regulatory sites within the DNA, are now possible. DNA sequence provides the opportunity to establish the relatedness of organisms, is illustrated by the RFLP analysis. But DNA sequence provides a much more detailed way to understand elements that organisms share and ways that divergence has occurred. Restriction enzymes provide other opportunities. If we combine digestion with restriction enzymes with DNA ligase, key in DNA replication, if you will recall, there are new opportunities that emerge. Restriction enzymes recognize specific sequences and generate overlapping ends, and that provides a special opportunity because the sticky ends of restriction endonuclease digestion can interact to form a semi-stable complex. And that complex can be recognized by DNA ligase to reseal the DNA to its original sequence. But remember, ligase is not sequence specific. It will reseal any nick in a DNA sequence. And this feature of ligase makes it possible to recombine DNA from different sources as long as the sticky ends match to create a nick. DNA that has been generated by restriction enzyme digestion and then inserted into another DNA using DNA ligase is termed cloned or recombinant DNA. And the sealed ends retain the original restriction endonuclease target site so that treatment with that enzyme will recover the inserted DNA. Now, plasmids are common carriers for cloned DNA. These small, usually circular DNAs are separate from the chromosomal DNA in bacteria with independent replication processes. They occur naturally in bacteria and occasionally in eukaryotic organisms. Easy to isolate, plasmids are amenable to insertions of DNA, creating plasmid combined with inserted DNA, which is called recombinant DNA. Because plasmid DNAs replicate within the bacterial cells, growing bacteria generates large amounts of plasmid DNA and expression of any proteins that are encoded by the recombinant plasmid. This method allows isolation of significant amounts of proteins for which DNA coding information is available. But the protein itself may be produced naturally only in small amounts. So this approach is one of the key methods of modern biological sciences where proteins even produced in small amounts in the cell can be produced in large amounts and characterized. One can take another step, and that is the introduction of coding sequences for proteins for expression into a eukaryotic organism, including people. The sequence encoding a protein can yield the protein in prokaryotic cells or in eukaryotic cells. Taking the next step, it is possible to introduce foreign genes into organisms using DNA transfer in order for those genes to generate a protein. The process is called somatic gene transfer for humans, with emphasis on somatic, and take just a moment to think about why. Somatic gene transfer is utilized where an essential gene in the individual produces a non-functional protein. A cloned gene encodes a functional form of this defective protein and is introduced into a cell isolated from the individual. The cells are grown and then injected into the individual where they home. They go to the organ type, blood or muscle, for example, to produce the missing protein for the person. So, when is this type of therapy utilized? A great example is for individuals who are born without the enzyme adenosine deaminase, which is essential for immune function development. Without an immune system, these children die very early from infection. This type of treatment comes with significant risks, and several deaths early in this process treating other diseases slowed progress in developing this approach for humans. But one of the aspirations in this area focuses on eukaryotic stem cells, where the ability of these cells to form any other type of cell indicates that a permanent change would be possible using these cells. Introducing modified DNA into these cells could provide relief for many medical conditions. This process is highly regulated by federal agencies, and prior impacts have included the development of cancer. The positive side is that tissue rejection may be avoided for transplanted cells if the original cells are from the same organism. Many researchers are motivated by the concept of personalized stem cells that can replace our damaged tissues. But we are only talking about somatic impacts. That is, introducing somatic cells does not affect the germline. In contrast, transgenic organisms are produced by introducing a DNA change into the germ cells, thereby creating a new organism with a permanent change in the DNA sequence. A foreign gene is introduced into the DNA, which is injected into the nucleus of a germ cell or an embryonic cell. The foreign gene is incorporated into the DNA of the germ or embryonic cell and is part of the DNA of all of the cells generated subsequently. The method involves putting a transgenic embryo into a female mouse to generate progeny, for example, that have different DNA and different features from the parent mother, as illustrated here. There are hundreds, perhaps now thousands, of mouse lines that are transgenic, some even sporting a humanized immune system. And many other transgenic organisms, from single cell to plants and animals, have been generated. Except humans. Transgenic humans are disallowed by law in most countries because the risks of the process are manifold, and the changes introduced are permanent in the genetic lineage. This issue may well arise in a variety of ways over the coming decades, and being aware of and knowledgeable about the issues is important as we move into the future. As you reflect, what makes the combination of DNA synthesis, restriction enzymes, and polymerase chain reaction powerful in such a wide variety of arenas? Why is the ability to sequence DNA so important? The capacity to alter genomes has long-term implications. Can you identify several of these? Should limits be placed on transgenic organisms, on transgenic humans? Why? Why not?